Okay, Bismillahirrahmanirrahim. Assalamu alaikum. Some core knowledge, some concepts that we cover in different chapters, including excretion, and then you have coordination and some part in homeostasis. I received some question that we need to cover some concepts here. So I'm sure that it's better that you also go through questions, but let's first start with the excretion. I think that was the easiest chapter, if you remember. The only thing you need to remember what exactly is excretory products. Is my voice clear and you can see the slides, the screen?
Yes, sir.
Yes, sir.
Okay, good. Okay, so remember that excretion is very, very important because we have ongoing chemical reactions going on in our cells. And if we, during that time, of course, it's needed for the cellular survival and for the growth and different functions of our body. So one of the purpose of excretion is to get rid of the waste products. You remember that glucose and oxygen constantly reacting, metabolic reactions are taking place, and that is generating carbon dioxide. Carbon dioxide, which is generated in our body, is of course taken care of by our lungs, right? So lungs can easily get rid of extra carbon through gaseous exchange. But what about those metabolic chemicals? Because they are poison, remember. So you have to be extra careful to get rid of these poisonous material if they are building on in your body. So that is extremely dangerous for our biological system. So one of the part that we discussed was in human is the kidney, right? There's the most excretory. That is the main organ which is involved in excretion of these toxic metabolites or toxic chemicals. So the major thing that we call it why kidney is producing urine, and urine is because of the nitrogenous waste, which is really dangerous because of the urea. When you say nitrogenous waste, it should contain nitrogen, right? Amino group in the structures. So from where we get amino acids, you need to think about proteins, because proteins are made of amino acids, and these amino acids are metabolized, of course, in our body by liver. So that is generating urea, okay, as a byproduct. Urea is, of course, a toxic substance. I think we discussed here, and we should think we can easily say that when we are ingesting protein, the digestive process, it goes through tract, right? So your stomach, your intestine is involved, and it's absorbed and breaking down the amino acids. Amino acids are absorbed. Protein is broken down into amino acids. So amino acids through the portal veins. Remember that whatever is absorbed through the intestine is going through the hepatic portal vein. This is very important because from the intestine, this is a safest way as well, because if some of the chemical that are toxic, before going to general circulation, you need to metabolize them, you need to detoxify. So liver is the main organ which is detoxifying. So during that process, of course, there are certain byproducts that are generated. So one of that is urea, okay? And I think we have explained it here, and in this figure is nicely shown here what exactly you see in this figure, that you have proteins that are broken down. The amino acids are going through the capillary portal veins. So these portal veins are going through the liver. You have amino acids. Remember, proteins cannot be digested. Whenever you think of protein, so the currency in protein is amino acids. Like we have starch, we are getting glucose, right? From fats, we are getting fatty acids. So we are getting amino acids that are now either processed for different proteins, because we have different enzymes, the cellular structure, and most of the body part that are in the need of amino acids constantly, because we don't store amino acids, right? So amino acids either is going through general circulation, which is going in the body, that is needed for different functions here, okay? So this amino acid is either released into the common pool, which is the for the general circulation, or there's two ways that liver is processing amino acids, right? So amino acids is the ammonia deamination, which is the removal of the ammonia part, which is further processed into urea, and the carbohydrate parts, because you remember that amino acid has got both carbohydrate parts and amino part, right? So carbohydrate parts might be used for as energy reserve. You should remember that from where the urea is coming from, proteins, and proteins are giving amino acids. Amino acids generating urea, and this urea is now in the blood. So once it is in the blood, you have efficient system. You should remove this urea because it's constantly generated. If you are eating, you have more amino acids. Some of them are used, and part of it is then converted to urea. And this urea should be removed from your body. And you think about what is the main organ, that is the kidney, and how kidney is processing blood, first of all, because urea is in the blood. Okay, so whenever you ask about urea, how it is generated and how it is processed, through filtration. You remember that what is the job of kidney is filtering the blood or plasma. Okay, so what exactly they have mentioned here, that uh, I think this we have explained it here in the part of amino acids. Okay, uh, and then it's they have given you different waste products and how they are generated and how they are removed. But I think the best part that we'll talk about is the human excretory system. Okay, and what is the excretory organ that we have in our body? Kidneys. We have two kidneys. Okay, so one can of course survive with one kidney if it's working efficiently. Yeah, but usually we have two kidneys, and how this kidney is uh, processing or removing urea from the blood through the, if you remember, the functional part of kidney is nephron. Okay, what is nephron, or the functional unit? These are small tubules, and that is, kidney is made of millions of nephrons, right? And as you could see here, that it has got medulla, and the dark part of the medulla you have Bowman capsule, where the filtration of blood will take place, right? So a loop of Henle, different structure, I think is shown here as well. If you look at the nephron, is a very specialized unit of kidney. Okay, so they have given you full structure. If you remember what is going on, what are different processes that are taking place in nephron, then you can understand. First of all, as you know, filtration is the most important part here, because you could see here blood is brought here, so it's constant supply, a good perfusion. And if you remember in exercise, we mentioned that the blood flow is also, you don't see much filtration in strenuous exercise, right? But when you are at rest or normal activities, you see that blood is constantly supplied, and this is called the glomerulus. This is the tangential or capillary network, and the flow of blood is is taking place in a very specialized way. As you can see here, the supply is more compared to the draining, right, to leaving the blood. Okay, so glomerulus has got much pressure. So this much pressure is responsible for the filtration, because it's squeezing the blood. So it's squeezing, and things will come out. The water will come out, glucose will come out, amino acids will come out, some salts will come out, right? But keep in mind that bigger molecules cannot be filtered. So, okay, until there is damage to the capillaries in case of high blood pressure or in case of diabetes. Okay, these capillaries might be damaged. So in that case, you might see excretion of bigger molecules. That's why you don't see proteins in the urine. Okay, urea is water soluble, it can be easily filtered, right? Glucose is water soluble, so it can be easily filtered, but glucose is reabsorbed. Majority of the thing that is going through, you can see the structure of the nephron in a different way, has a got very specialized function. So you also could see that they're wrapped up with the capillaries, and these are veins that can reabsorb. These blood vessels can reabsorb things from the nephron, things that we need, like glucose are needed, water is needed, and amino acids are reabsorbed, sodium, electrolytes, and other things are reabsorbed. Urea is not absorbed, urea is taken out. So that's why we call it urine. So if you remember what is urine, is a solution of salt in urea, because urea that was generated is in the blood. So this blood is easily filtered here. So you can see here filtration, they have nicely explained here the exact mechanism that I mentioned, that you have high pressure here, so supply is good then compared to leaving it. So you have high pressure here that will squeeze the blood out. So that's why squeezing would cause more things to come out of with water, right? So those things that are soluble. So this is also called Bowman capsule. The cortical region of the kidney that we mentioned, the dark in color, or these are so many capsules that you see here in this medulla region, right? And then coming back into into the cortex. And remember that all these nephrons are now finally pouring this filtrate into pelvis region, and pelvis is now connecting again into bigger tube or duct, which is called the tubing is the ureter, and this ureter is finally bringing these to the bladder. Okay, and then I think we have seen it here that each kidney is bringing filtrate to pouring this filtrate into bladder. Okay, in this bladder you have two big ureters here, and then there's a sphincter. This sphincter is controlling. In this book they have mentioned that this control is not attained until age 8 years. That is why newborns and also in some disease condition they cannot hold urine. Okay, it will automatically pass, but we can hold a volume of urine. Of course you still have a capacity to stretch, so this stretch depend, how much urine we can hold. So adult, maybe they can hold a good amount of urine until you have urgency, and then you have a signal which is going to the brain, and they will definitely going to open the neck, and then the urine will pass through the urethra. So this is under the control of higher center. But as I mentioned, that it's involuntary. They cannot control in very small age, and some kids they may even till age 10 they cannot maintain. This while bedwetting is more common in such children. They cannot hold urine, right? Or due to surgical procedure, due to radiation and any other disease condition, people have incontinence. We call it incontinence. Okay, I think they have already mentioned here. So I think you remember the mechanism of filtrate. Okay, the capillary network, the glomerulus. You think about this is the main thing you should remember. Why glomerulus is quite wide, the blood which is reaching, and they are filtering the blood, the plasma. Okay, so when the filtration is taken place, okay, the blood is filtered, the next process is reabsorption. Okay, because we may preserve things that are needed by the body, especially the glucose, as I mentioned, amino acids. Okay, the urea which is dissolved should not be absorbed. Okay, that's not absorbed, so that is taken out of the body. And this reabsorption is because of the blood vessels, right? These vessels that are surrounding these nephron is a very good mechanism for reabsorbing most of the things. So what is reabsorbed? Your glucose is reabsorbed, amino acids are reabsorbed, and then I think they have mentioned the glucose, amino acids, yeah, they are reabsorbed. So filtration and reabsorption. Okay, so you can see here they have mentioned in this figure the whole process of filtration. Only small molecules such as water, glucose, salts in urea are squeezed out. Okay, and then reabsorption is the water and glucose are taken back because we want to restore. And this is how your body is making urine. Okay, this whole filtration process is making the urine, and they have given the definition of urine. If you are asked what exactly is urine, you can easily guess the name: urea and salt and water. So this guy urine, urea and water is basically urine, right? It's acidic in nature. I don't think you have any other important aspects here. What is here? The remaining liquid that is going out is called urine. And I think in one of the questions that you mentioned, hot weather or cold weather, you mentioned about that. Although that was talking about the salt overall, one question you shared in the group. So your kidney is taking good care of water. It depends what kind. If you are doing strenuous exercise, of course you don't want much urine to be produced. You want to preserve water because you have sweating, and that sweating is also you're losing much water. In winter, for example, there's no sweating, so that's why whatever water you take, that is efficiently filtered. You might see people who are sitting in air-conditioned rooms, or in case of winter, if they're taking water, they definitely have more urine output also. In hot weather, where there's a lot of sweating, your kidney is preserving water because you don't want water to be lost by both systems. sweating, and that's also one of the principle of homeostasis that we have mentioned, and under the control, how much water you want to preserve. Okay, so reabsorption, you remember, is a simple passive way, and things are easily dissolved. These are lipid soluble things, and or maybe small molecules to the water channels and getting back into the blood. So
I think sir left.
Yes.
Okay, I'm sorry. This I don't know. Some Now I think let me continue where exactly we left. So you remember that we talked about the main function of the kidney and then filtration and reabsorption. And I think you usually remember what exactly is bladder. I mentioned before the capacity is just to hold the urine and then passing through the urethra out of the body. Okay, so this is again depending on how it is controlled. For example, adult mammals and can consciously control this sphincter, the sphincter that I mentioned before here, under the control of muscles. Okay, these are basically some muscles, and we do have some diseases that may affect this capacity. There's some people they cannot hold urine, or maybe they cannot pass urine very easily, and that's why we give this drug to give the sphincter muscles. There's the opening in the bladder, there's the opening of bladder through the urethra, so this sphincter which is open, and then that will pass the urine through the urethra and outside the body. So dialysis, I think, is a very special need of some patient, those who cannot do well with their kidneys. So their kidneys are not functioning properly because remember the most toxic thing that we need to remove through dialysis is urea, although we have other electrolytes also. So this is also called artificial kidneys. We also call dialyzing machine or kidney machine, or they are filtering the blood the way your kidney is filtering. Okay, of course it's not under control of central nervous system the way it is preserving. It's need a lot of control. You cannot leave it just like that. Your kidney, mashallah, is doing perfect job to control most of the things, so it has a lot of limitations. So the thing is that you consider this part, that you should remove the urea and other products, other toxic chemicals from the blood outside the body artificially. That's why they are connecting this dialyzer. This got dialysis machine, and you have dialyzing fluid. This solution is containing those things that you want to retain. You want to retain that in the blood. Okay, so the things that you want to remove should not be there. So it's kind of a whisking tube that may allow part of things, and not allow all the things. So of course it should not contain urea, because what happens basically, you are taking blood. This is the this is how this the inlet, and this is the outlet. Okay, so this blood, you have a small shunt or uh, another connection, and that's required because you want enough amount of blood to be pumped through this dialyzing machine. Okay, so this dialyzing machine, these are kind of a whisking tubes. So that's why they are saying that inside this blood, part of chemical, that for example urea, will be taken out of the blood, because the blue part, the background that you see, this is dialyzing liquid, a solution. Okay, so it does not contain any urea. So urea has less in concentration, so it will come out of the blood, and it will be, and you can see here that this dialyzing solution is now taking out. Okay, so inlet, and they are saying that dialyzing fluid, the speed is, this dialyzing fluid should come with little less speed compared to the blood. The blood has got more pressure. And if you remember, what do you think this is resembling somehow glomerulus? How do you relate this explanation with glomerulus? What exactly was needed for the things to squeeze out, for the glomerulus to squeeze out things, and the nephron? Can you relate somehow?
Yes, sir.
Yeah, keeping the function of pumps. Would you like to explain a little so that everybody understands?
Sir, the high blood pressure helps it.
Okay, they're saying the pump is keeping the blood moving through the dialyzing at a higher pressure than the dialysis fluid. So the fluid here is less in pressure compared to the blood. Okay, any special reason about that?
Sir, partially permeable membrane.
Partially permeable, but if we keep the speed same, would the dialysis be efficient?
Yes.
Uh, Okay, then why they are putting this pressure of blood higher compared to dialysis fluid? So the fluid which is coming through this way, this is also special filter, okay? And you also have a special filter for the blood as well, because you don't want foreign particles, anything should go in your body, because that is extremely dangerous. You cannot introduce any foreign particle, insoluble thing should not go in the blood. This is one of the reasons that filter should be there, okay? But I'm just asking about the speed of the dialysis fluid and the speed of the blood. Why it is different? If we keep it same, would it make any difference? Anybody would like to give a guess? What comes to your mind? So remember, which part is removing the toxic material from the blood? Dialysis fluid, right? So dialysis fluid should have some time to have contact with blood. Blood should definitely move faster, a little high pressure, because you need that to squeeze out the thing. Yeah, you basically need it, because and this will somehow collect, like it will take the urea, and this urea is then now removed from your blood. So that is why this speed should be lower. Dialysis fluid, it should be lower, and it contains glucose, sodium, and potassium. You don't want glucose to be removed from the blood here, much of the glucose. You want to retain it. You just want to remove those things that are not needed, especially urea. That is why dialyzing fluid should have less urea. You might be given some question with different concentration of urea. So keep in mind that the basic purpose of this dialysis is to remove this toxic material, urea. This is why you should not introduce urea in the dialysis fluid, right? This is very, very important. And why we make a shunt, which is alternative path, a special connection here with the artery and a vessel, so that you can have more blood to be filtered. You have more amount. You also provide collapse. You also avoid collapse of this vessel if you don't have much amount, because you want to provide much pressure here, and that is required for the dialysis. And why we need dialysis? In some cases, if there's infection, maybe the kidney stopped working. Uh, if this is one way, if patient can survive with dialysis, but keep in mind that you need to repeat it couple of times, couple of sessions in a week or month, depending on condition, uh, how much is the impairment. If the renal impairment is huge, you might need more dialysis sessions, and it's very really expensive as well. You need to admit the patient and all the come. It's not that easy, to. So people sometimes definitely go for kidney transplants. So a dialysis machine, do you think there's something special they have used? I think this is the whole mechanism, right? So the fluid waters plus salts and other substances that should be present in the blood are there in the dialysis fluid. You don't want to get rid of that. Is that clear?
Yes.
Okay. So coming back to transplant is really needed in those patients who cannot do well with dialysis, with artificial kidneys that we mentioned. So the final option is transplant. You remember, since you are getting organ from a donor, so somebody has to donate that kidney. You might have seen people are donating one kidney to some people, to the close relatives, or people are also carrying tags that they would be willing to donate their kidneys in case sudden death or due to accident, for example. So but that has to be removed immediately, okay? The kidney has to be removed immediately, and it has to be maintained in a very special condition, okay? So you are basically removing that organ for transplant, so you remove it very quickly, otherwise kidney will die. Or you keep it also in cold. You don't expose these cells to be deteriorated or damaged because of the excessive heat or not favorable environment. And once the kidney is transplanted, remember that this is a foreign organ, so your body is not familiar with this organ, right? So your body is not familiar. That is why you need to have immunosuppressant drug. Immunosuppressant drugs are those drugs that will suppress the antigen antibody reaction, because your if somebody has received kidney and his own immune system is attacking the kidney, because the immune system is not used to with this system. That is why the person who is the donor, if he's closely related to the patient, so you probably have good genetic matching. Okay, the match will be really efficient and very good. But if it's taken from distantly related, so definitely genetic differences are more, so your body will start reaction against this kidney, so that transplant could be rejected. This is called rejection. Rejection means it will stop working and you have to remove it. So the organ is cannot stay in the body, you have to immediately remove that. And that's why to avoid this rejection, we give them immunosuppressant drug. Immunosuppressant are those chemicals, those drugs that will prevent or decrease the chances of rejection, because these are suppressing the immune cell. But at the same time, keep in mind, when you are suppressing the immunity, what will happen? So these patients will also suffer other sorts of infections, diseases, because you are basically using immunosuppressant drugs. Okay. So although the chances of rejection are reduced, but at the same time, you expose this patient to more infections. So these patients may not be very good in immunity. For example, in these days, if somebody viral, where corona pandemic is very common, right, everywhere, so they can easily catch those infections, and these infections could be extremely serious in such patients because they are taking immunosuppressant for a long time. So immunosuppressant, apart from immunosuppressant action, which is maybe needed to avoid rejection of kidney, but at the same time, those immune cells that are needed to fight against diseases are also uh, eliminated. That part is also eliminated. So this is really not good. That's why the chances of rejection would be reduced if you have close relatives, right? Because this is called likely to have antigenic properties. Antigen is the kidney. Yes, Muhammad Azam, you have a question?
Yes, sir.
Go ahead, please.
Sir, are there any chances in future that they stop rejecting the kidney?
Is there any chances to stop what? Sorry.
That the immune system stop reacting to the kidney?
Usually they start acclimatization. They may call it that probably your body would start accepting. Initially, you might need very high doses to suppress this immune reaction against kidney, but usually then your body is probably accepting that. So it used to, you may not, although it's usually long process, chronic treatment is needed. You still would need some immunosuppressant drugs to avoid rejection. You cannot totally rely on body antigenicity, because this antigen is not efficiently related or not closely matched. But at the same time, it may get used to it. It will start accepting that antigen. So you gradually, definitely the chances of rejection are decreased initially. That's why a rejection is mostly common in initial phases. If that is successful, inshallah, it will also go well in the later stage of their life. Is anything that I have missed here, you think, or we have covered the things?
No, sir, I think we have covered it all.
Okay, good. We have covered that all. So you remember glomerulus, you remember what is filtration, you remember reabsorption, and you have mentioned about dialysis. Dialysis, of course, is even not easy. As you know that even cost factor is there, and chances of infection, because you are taking the blood, it's not efficiently, you could say, protected the way it's protected in your body. So the blood is now outside body. So chances are that you might see introduce infection, and if there's some problem with the filter, you might introduce some foreign particles. So they may not be really good for the patient, right? The same is the case with the transplant. In one hand, of course, you are giving more new kidneys where they can work efficiently, but you have to maintain that patient on immunosuppressant. And this is again, a risk is there. Anytime it could be rejected due to antigen antibody reaction, and even if it is not rejected, the chances of infection are more. These are some of the disadvantages of kidney transplant and dialysis. That's why your people are those who are obese, those who are diabetic, those who are hypertensive, to have good control on diabetes, to have good control on diabetes, okay? To have good control on hypertension. If your doctor would say avoid salt, avoid sweets or other carbohydrates food, so better do it because that will protect you in long term. Yes, Muhammad?
Sir, can the kidney be taken from a dead person?
Dead person, but immediately. Those who have, that's why they carry, in Western countries they have already registered their name. If due to accident they are dead, and immediately they are allowed, I mean, they remove the kidneys and it can be transplanted. But if you delay it, it's of no use. It's of no use because the cell definitely will not survive. There's one of the things that you need to mention immediately. It should be immediately removed, and then it's a special time there, and then you have to keep it in a special environment. When you are transporting that kidney, of course you don't have patient right at the scene, right? Maybe accident went happened somewhere 50 or 100 kilometers away, and you are transporting that kidney maybe 3 to 4 hours there. So you need to then immediately remove it and maintain it in a proper temperature. And they have instructions actually how to maintain and how long you can transport that. This is important, okay? The next chapter that we'll talk about is coordination and response. Whenever we talk about coordination, you always think coordination of your different systems, your body gesture reacting to the environment, your external environment. If temperature is too high, temperature is too low, you are touching hot surface, for example, you are pricked or bitten by some sharp object or some insect, for example, you immediately withdraw your hand. That is the response. This coordination is very efficient and very quick. So whenever we talk about coordination, don't think about the stimulus, okay? So you remember we talked about here stimulus, okay? We call it stimuli if for the plural, and stimulus is one. And then you have the receptors, the stimulus which is received, and then executed by effectors. So stimulus, receptor, effector, triangle. This triangle is very important, okay? You have a stimulus, you have the receptors to receive that, and then you have a proper communication to be executed, what to be done, in case your organs are involved, for example, muscles or glands. As I mentioned, when you are climbing upstairs, you definitely see your blood pressure. And if there's too cold outside, your body has to react to that stimulus, right? So if there is any damage to your body, hot surface, you touch a hot surface, what happens? That is why this process should be in a very fast and quick way. That's why we have very specialized cells, okay, the nerve cells. The nerve cells are really very, or we call neurons, are very much specialized cells. And you could see they are different from muscular cell or your kidney cells or your heart cells, for example, cardiomyocytes, or muscle cells or scans, for example. They have very specialized dendrites. You can see here the structure, and I think you need to remember the difference between what are different kind of neurons, what they are doing. They are very specialized, and their cytoplasma is extending, right? So this is called the axon. This is the extension, and you could see myelin sheet are covering this part. So if signal is anything is received, that is propagated through axon very quickly. And then you have also a gap, and this is the myelin sheet and node of Vanweer. And you remember what is the function of this gap and what is the role of myelin, okay? So here the main thing that you should remember is the body of the cell that is receiving dendrite. You can see here these projections. They are very good at receiving the signals, and then is passing through the axon. An axon is special. A myelin sheet is a proteinaceous kind of a covering, and if you don't have myelin sheet, the impulse transmission will be not very fast. It will not be very good. I think they have given you an example here somewhere. Look at this. If you have myelin sheet, so it can carry impulse with 100 meter per second. Now remove the myelin sheet 5 meter per second. Do you think 5 meter per second would help you to give a quick response? If somebody is immediately stepping out of the road, jumping, the heart rate should immediately increase. Your circulation should immediately increase. Your respiration should immediately increase. the moment you jump, all of a sudden you see the transmitters are released, and they are doing this function very quickly. And that is how your brain system, your central nervous system is designed. If there is any delay, you can do a lot of damage to that organ. For example, somebody was sitting, and all of a sudden something appeared, maybe snake or some dangerous or scorpion. So you are scared and you jumped out of the table, or you are sitting. When you are sitting, your blood pressure is very low, your heart rate is low, your respiratory rate is really low, because you don't want that much energy. But the moment you step in, you just jump, there should be immediate increase. If you don't increase, for example, heart rate, what will happen? Person will fall down. He will fall down, fainting. We call it fainting. The person will immediately have syncope, immediately cannot maintain posture. It will just fall down immediately, because you don't have enough supply of blood to the brain. Your brain is coordinating, your muscles cannot work properly. That is why this system has to be efficient, and this efficiency is because of these specialized cells that we have. Okay, and these specialized cells, you can see the structure is also very special. And these are nerve endings. What are these nerve endings? So these neurons are communicating with each other. These nerve endings has got vesicles, and these vesicles are storing transmitters, and we call it neurotransmitters. These are called neurotransmitters, and these neurotransmitters are released. There's also another cell body of the another neurons or nerve, and that will call we call it synapse. Okay, synapse. When there is a space, a junction, and this junction is not in direct contact with each other. This nerve ending is now connecting with another cell body. Okay, so transmitter are released, and they are working on different receptors, and these transmitters are also innervating different organ. For example, in case of muscle, if transmitters are released in the muscular region, in skeletal muscle, your body, so you immediately withdraw your hand. This contractions is stimulated by these transmitters because of the nerve ending are releasing. Okay, so then you need to remember what are sensory neurons, what are motor neurons. When you say sensory neurons, that are carrying information, okay, sensing, they are receiving hand, they are receiving the information, and then is going to the higher center spinal cord or brain, and that sensory are taking impulses to the relay, and relay neuron, depending on the response what to do, they through the motor neurons they are propagating or executing the whole activity, and that is under the control of central nervous system. Okay, so whenever you talk about the neurons, don't forget about myelin. Myelin sheet is extremely important. If there is a demyelination, and then we have diseases. Okay, we will not go into details, but yes, this transmission is not going to work. And people who are in old age, those people who are drinking alcohol, those who are smoking a lot in the old age, definitely they have these myelination problems, so they cannot coordinate activity very well, and people may die also because of this lack of coordination. So I think your central nervous system is coordinating, you can see here brain and spinal cord. Okay, because these sensory neurons are providing these information to your higher center, and this is the control, like exchange of your you could say the CPU of your body, the computer system that you are through the keyboard you are inputting information, giving information. Once this information is then analyzed by your higher center, what to do in case of reflex arc, which is a very good example of coordination. Okay, so I think they have given you different neurons here. So you could see here in this, for example, I think we had few questions, you remember, in that, and they were talking about the sequence. So whenever you're taking, they might give a labels here, like this is P, and this sensory neuron is going and taking the impulse here. Okay, so you can see the direction of the arrow. Okay, so don't confuse that, which is to the organ, that is to the motor neurons, because that is executing the function. Okay, so don't confuse that with the sensory. Sensory, this is how your process, the reflex arc, will originate or initiate this response. Okay, if there is no stimulus here, you don't have any reflex arc, right? If you touch anything hot or maybe pricked by a sharp object or pin, for example. So you start from here, okay, and then you see here the flow, and then the relay neurons, and finally bringing execution to the muscle where you can easily withdraw your hand. And they have given you a very good flow here. I think you could easily see reflex arc flow here. This is the sensory neurons, because you have received the stimulus, okay. So the stimulus receptor, and now the whole communication is now going through sensory, through relay, and then finally to the motor neurons, which is executing the function through the effector. Effectors are your organs, okay, your glands also. As I mentioned that uh, when you are eating, for example, sweet, and your body has detected more glucose in the blood, so insulin has to be released now. So how do you know that this is not under your control? That is under your higher control. Okay, so it's going to give signal to pancreas, and it's releasing more insulin, and the glucose is taken good care of. But if someone has problem with pancreas not releasing insulin, so definitely this is deficiency of insulin. That's how we have diabetes. So diabetes also, in case of homeostasis, this is already controlled. Homeostasis and coordination response, you can take it together, because they have close link. They are, most of the principles that we will discuss here are also related to the homeostasis, because your brain system and your receptors and the signal transmission and in the neurotransmitters are communicating. The neurotransmitters are the just like electrical signal. This is called the electrical impulse. Okay, electrical impulse, we call it. The stimulus will generate electrical impulse they are releasing different transmitters. So I think reflex arc. Is this part is clear to you guys? The reflex arc, you won't be able to confuse sensory and motor neurons. I remember somebody started the flow from here. I can't hear you. Somebody's talking. So don't start the flow here. When we say motor, that is executing. That is connected to the effector rather than the sensory. Sensory, you can sense. Your sense, you're receiving the stimulus, okay? And I think this is extremely important to understand what are different neurons, okay? The types that we have. So you have this one is the sensory one, you have a motor one. So the cell body, you can see, is the main difference. They might ask what exactly is the cell body is away from ending, while in case of motor neuron, it is at the one end of the body, right? So in case they have short dendrites, the relay neurons is interconnected, okay? So sensory neuron is propagating this impulse to the relay neuron, and relay neuron then finally propagating its action through the motor neurons to different glands or to organs like muscles, for example. So I think the most important here to understand is the synapse, right? So what is synapse and what is synaptic cleft? That is another topic that you should understand. How they are communicating, these neurons. Is that the functional unit of your brain is neurons. The way you have nephrons in the kidneys, right? So neurons in the brain, in the higher center. So they are communicating, they talk to each other. There is a crosstalk, interplay of they are communicating to each other, okay? And you can see that these neurons that are involved in the reflex arc that we mentioned, they are not connected to each other. They are not in close contact. There is a small gap between each pair. Okay, the gap is called synaptic cleft. I think they have given you example here. Okay, so you can see here, this is the nerve ending, and this is the another cell body of the next neurons. Okay, so you can see here that there's a small gap. This gap, narrow gap, is called cleft, synaptic cleft. In this whole pre and post synaptic is called pre and post. Okay, so the one bring is either before the synapse or after the synapse. So when they are the signal is traveling through this axon and it's reaching the ending, nerve ending. So these vesicles, these vesicles are storing these transmitters. The transmitter are released. So now this impulse, the stimulus is now transferred, or this information are transferred to the next neurons. Okay, so it will carry, because some transmitter were released, and this transmitter are under the control of this impulse. If there is no impulse, you won't see any release of transmitters. If I am picking this pen, for example, and the moment I want to extend my hand, so of course I can pick it because I can contract the muscles now, because my higher center has released the transmitters. And now if you disconnect somehow, if you paralyzed by the local anesthetic, right, you block the transmission, okay, nobody could pick it. Why? Because this hand is now not connected with the brain, so the transmitters are not doing its function. You have blocked the transmissions. You have blocked the release of transmitters. They block the electrical impulse. In case of polio, for example, there's also disconnected. That part of hand is disconnected. So you have the blood supply, you have everything, but you don't have sensation. That is why even if somebody will put it on a hard object, so they won't receive any impulse, because it's not. Similarly, the taste part is also a very good example of the coordination. I think so. Now you're clear about the synaptic lift, and they have given you this figure. So combined pre-synaptic neuron, post-synaptic neuron in the whole space. This is called synapses. This is called synapse. In the cleft, the space is called synaptic cleft. In this cleft, actually, you have transmitters that are released. Is it clear?
Yes, sir.
Okay, let's talk about the receptor buds. I think these are again the sense organ: your eye, your skin, your hearing, your touch, your smell, and also the taste buds, for example. Okay, do they have anything? Right, let me see here. So the next organ that they are discussing about the control and coordination is a very good example is your eye. In your eye, you have to first of all remember the structures of eye before we talk about anything. Okay, you have the cornea, which is the window, the upper transparent region, and then you have the black hole that you could see. That's called pupil, and the colored part of your eye is the iris. And iris contains the circular muscle and radial muscle. You remember that iris is sensitive to light. Okay, so remember what basically your eye is doing. Whenever you want to see an object, you are receiving rays, right? These rays are passing through cornea, through the pupil, through the lens, and finally in the retina. So retina has got these where the image is formed, and this retina, of course, you have the optic nerve which is carrying these impulses into the brain, and how the image formation will take place. But your retina is highly sensitive to light. Your retina is very sensitive because it's like the film where the image is formed, right? These all lens and other things, they are just controlling the entry of light rays. So you want to bend more light. So if you looking at a near object, okay, your lens should be a little bit thick enough, because you are receiving a lot of rays from distant, from closer place, right? I think they have shown it here or here. So if you're looking at the near object, this is the near object, a lot of rays. So you need more efficient banding of light because you have receiving a lot of rays. But if you're looking at the farthest place, the rays are coming from distant, they are traveling in parallel direction, right? So this has to be thin. So this is again controlled by these muscles that you have in the suspensory ligaments, and then you have the iris. Okay, so let's talk about first of all the retina. And the retina has, not the whole retina has sensitivity to light. This fovea has got most of the cells, and these cells are rod cells or cone cells. Okay, and depending on which color of image you see in the dark or you see in the light, if you see the colors that you see is because of the cones. Okay, the right is only in case of dark, okay, black and white image, not the color one. So this fovea where you have the sense, light is this part is sensitive to light. Okay, you can easily control that. And whenever you have too much exposure of light, you don't want your retina to be damaged. Okay, so that is why you would see the iris, these radial or circular muscles. Depend, if you are looking in the dark, you need more light, right? But if you're looking in a bright light, you don't want to enter much light here, okay, because your retina is sensitive. So this will control the entry of light, okay? So the entry, that's why your circular muscles are contracted. And if you look in the mirror and somebody all of a sudden put torchlight, you immediately see that the black dot that you see in the eye will be constricted, or this is called constriction, so that it can limit the flow of light. But when it is dark, all of a sudden it has to dilate. That's why you don't see for a while. If you are in a very bright room, in all of a sudden you switch off light, so this has to adjust. You cannot see things all of a sudden. This has to adjust. The people side has to dilate a little so that you have more entry of light, and that's why you could see things in the dark. Okay, but you can see here, you could just see the shapes. But if you have switch off the light, you may not be able to recognize which color is it, red, green, or yellow, or so the color part is basically taken care by the cones. And I remember, if you forget which one is responsible, C is for color. Okay, C is for color, and they're responsible for cones. Okay, so I think this we have discussed, retina, and then yeah. So always read about. I think we already have discussed this in our lecture. You could also see the recording there if you think detailed discussion here. But since we have multiple things to discuss in this chapter, especially a few more topics that we need to cover, because you have exam coming up. So I'm sure that you remember here now, and you could see chloride is a black one. You could see that is avoiding the spreading of light. Okay, you don't want this light to spread in the other part. And the sclera and also conjunctiva is the covering part, you know, the upper cover with the blood supply here. Okay, and I think you remember here, and these are the you could see the conjunctiva here, and you could see the cilia muscles. So this is also contraction and relaxation would depend how much light you want to bend. Okay, if you're looking at a closed object, so this lens, do you think is for the closed object, the shape of the lens, or for the farthest object, or for the far away?
Far away.
This is for far away. Why?
Sir, because it is long
It's
and
oblong. Okay,
maybe it's
so what is the function of the lens?
to adjust the light going in the eye.
Oh, okay. Do you think adjusting the light is the function?
Sorry, sir. Like changing the angle, reflecting the.
Bending of light, yeah, to focus it at the region where it's needed. Exactly. So the light entry is controlled by your pupil, iris size. Okay, iris is constricting or dilating the pupil, the black dot that I've seen in the center of the eye. So that is controlling the entry of light. This is responsible for bending. When you're looking at the object far away, you don't need much bending, because you know whatever light is entering can be easily bended and could be bent towards the retina. Why? But if you're looking at a closer object, you have very close, you know, a lot of light entering, so you need this lens to be thick. So this is probably for the closer, nearby object, not for far away object, because you want to bend light when it's really concave and it's thick, not thin. In that case, it can efficiently bend light towards the retina. Okay, so I think this I have coordinated here, if you remember, with this figure. So you could see here, this is the distant object. You could see here the lens is really thin. Now for the near object, the lens is really poor, and also the ciliary muscles will contract, and that's the causing the lens to bulge. Bulging of this lens would be able to bend more light, to reflect more light compared to the one which is thin. If you're looking at far away, you might see they are going straight, so you don't need much bending. But at nearby object, you may see rays are coming from the closer object. So you need this wider area of the lens to cover and bend the light. This is one of the things. And I think they have also mentioned here about these accommodation. This is also called accommodation, when your lens actually is how it is. Yes, Nabi?
Sir, I have a question regarding the central nervous system. So there are two nervous systems, right? The one central nervous system and one peripheral nervous system. So can you explain the peripheral?
Yeah, the central nervous system is the one that we are looking at, the higher center. So the peripheral nervous system, which is executing, the autonomic nervous system, the organ system, the coordination system that we discussed. This is related to a periphery. It's not your higher center is your brain, okay, your brain and spinal cord. So whatever else, like you're controlling your heart, controlling your muscle, controlling the hormone release, is related to the peripheral system. Okay, so in the periphery,
Okay,
periphery
sir.
in the side. Okay.
Okay, sir.
Yeah, accommodation, I think, is another, is focusing of light is called the accommodation, right? What do you think? What is the how we accommodate to different rays, to the objects, the accommodation? Can you see here in this region how your lens is responding and how it is responding to different object? If you are looking at the faraway object or you're looking at the near objects, so that is actually if it is becoming bulging or it's becoming thinner. So that this mechanism is called accommodation, and it depends on, as I mentioned, that if the rays are coming from far away, you might see parallel and you may not see. Cornea is also bending some part of the light, and then lens is bending the light. Okay, but if it is coming from a very near object, it has to do a lot of work. So most of the rays would enter, and that would require bending, okay, towards the retina. So light is focused on the retina. So there's why the cornea is also bending the rays, and the what they have mentioned here, the thick lens bends the light rays greatly. They're saying that because you're looking at the near object, so there's why you might see that is really very good. The thicker or bulging of this lens uh, will be more efficiently bending the light and focusing on the retina, and this response to light, different, whether you're looking at the faraway object or nearby object, is called accommodation. This is called the accommodation, the difference, the light which is coming from distant object or nearby object, and your lens is responding accordingly. That is called accommodation. Okay. And this accommodation, they are saying that is done by, the part of this is the by suspensory ligaments that you have. So I think they have shown it in the next object here. In this figure, it is very nicely explained. Okay. You could see here that when you are looking at the distant object, the ciliary muscles are relaxed, and the suspensory ligaments are pulled tight, and this is one of the reasons that this will pull or make the lens thinner compared to the object which is nearby, where the ciliary muscles will contract and this suspensory ligaments are relaxed. So this just like you can say opposite, when the ciliary muscles are contracting, so the suspensory ligaments are slackened. When the ciliary muscle will relax, so this will pull tight. Okay, so for distant object, you could see the ciliary are contracting but suspensory are relax. Okay, so this is one of the things, this is tightened, so that will pull the lens to become more thinner for distant objects. So don't forget about this. If you're given a diagram like this, you could easily see, even by just looking at the shape of the lens, you could see, because this one is coming from the nearest object, so you need more light to be bent towards retina. Okay, the way they have shown it here. See here, the more light is the closer, so you need more efficient, you need more surface area of the lens compared to the one which is coming from the far away place. So this is one of the things that you have mentioned. I think one of the questions they have mentioned, which part of the eye is sensitive to light, so that is retina, because you have those cells rod in cones, okay, not the iris. Iris is just in response of retina. Retina want to be protected because you have the optic nerve which is providing these impulses to the higher center. So the higher center then will give direction to the ciliary muscles to constrict the pupil size or dilate the pupil size. If it is in the dark, that will dilate because you need more light to enter. But if it is in the bright daylight, so you need to control the entry of light to avoid damage to retina. Okay, so that is called one of, if you remember, the system that we mentioned was the either muscles or glands. So one of the things that we mentioned is the endocrine gland that is also responding in the coordination. If you remember, these are hormones that are released. So they are not just like chemical neurons, or they are releasing impulses and that is propagated to sensory or motor neurons, but they are releasing, these glands are releasing hormones in the blood. Okay, so when these hormones are released, so they are taking care of the activities of what exactly would be needed. For example, these hormones are then working on different organs, for example on the heart or in the kidney or in the vessels, depending what will happen to your body system and when it is needed. Okay, so that's called endocrine system. Endocrine is the hormonal system. Okay, it's not the coordination with the impulse transmission. Okay, here of course you have a stimulus, as I mentioned. For example, one of the best example they have mentioned is adrenaline, because adrenaline is the hormone which is released through the endocrine glands. Okay, and as you mentioned, adrenaline is again we also call epinephrine. Epinephrine, it is about the kidney, adrenal gland in the kidney, so that is releasing adrenaline. Adrenaline, another name for adrenaline is epinephrine. Epinephrine, nephrine is nephron. So about the kidneys, so adrenaline is released for fight and flight mechanism. So fight and flight mechanism is extremely important to respond. Your body should be quick enough, okay? Because when you see something, uh, maybe some danger or any scary situation, so what will happen that your brain is now ordering your adrenal cortex, adrenal gland, to produce more adrenaline. With the more rushing of adrenaline in the blood will bring what? I think you may describe your situation yourself. We are most of the time, especially people when you are in the classroom, some teachers are really very strict, so they don't like question. For example, all of a sudden people are scared, oh, I don't want to ask question, and their heart is throbbing, and then all of a sudden they are shaking, all the thing. Or you are in the park and all of a sudden you see police enter, and you see the police immediately, oh, there's something danger, maybe they're going to catch us, or maybe some activities going on which may not be safe here. So then you are reacting to this environment, you may be running away from their situation, so your adrenaline is now more released in the blood. Okay, so what adrenaline will do to your system, what do you do, how do you react to that situation? I'm sure that you can easily describe all the situation that they have mentioned here, the mechanism that they have mentioned here. So here the blood vessels in your skin will what? You remember somebody who is scared, the skin color become pale or whitish, pale, okay? Because you have much adrenaline. Adrenaline will constrict the vessels, blood vessels, okay? So first of all, the flight mechanism, the blood vessels in your skin and digestive system contract, so that there is very little blood going on the skin. Okay, this make you go pale and give you butterflies in your stomach, like your strange sound that are because of the muscle contraction, and also because of the less blood supply. A much blood is now possible, is needed for your brain and for the muscles to energy, because you want to prevent, you don't want your blood to go into digestive system, you want this blood to be diverted to the muscle and brain, because these are the organ you are going to use. Your muscle needs more energy. Okay, your muscle need more energy. So adrenaline also causes the pupil size of to go wide, okay, so that you can see very easily, you can see the near object very easily. And sometimes people are really they cannot focus much, and they may fall down. When they are running, they can just trip on things very easily because they are not focused, their eye is not focused, but their pupil size is dilated. That's why it's one sign also for the fight, especially in animals. When animals are angry, they fight with each other. This is one defense mechanism, that other predator or those they're going to attack will be scared because of the eye size is very dark and black, and you would see that uh, the dark circle in the eye is bigger, so they can move away danger, especially these animals can, the birds, they have this defense mechanism. We do have this mechanism, we do in case of anger. When somebody is extremely angry, ready for the fight, you might see the pupil size is very dilated. So this is giving because of the adrenaline. Okay, adrenaline causes the pupil size, and this allow more light into the eye, which might help you to see the danger more clearly. This is one thing. You want more light. Okay, you want more energy, especially blood flow to the muscle, so that you can jump, you can run, and you can use. You don't see easy fatigability. You don't get tired or exhausted all of a sudden. Okay. So, and it also to the liver, adrenaline will also release glucose into the blood. Adrenaline will from the liver will increase more glucose, glycogenolysis. They say that they will break down glycogen store. You have more glucose, so you have more energy as well needed. Okay, so these are some of the function you could see they have mentioned about these hormones, but adrenaline is the most. So adrenaline is released from adrenal gland. Okay, and that is preparing you for fight and flight mechanism. Is that clear?
Yes, sir.
Okay. This one, I think you can remember, they are preparing body for fight and flight mechanism for rigorous activities, also rigorous action. I told you, running, jumping. Okay. And then you have pancreas, you have insulin also, which is reducing the concentration of glucose in the blood. Okay, because it's storing glucose in the liver to glycogen. And also in case of the sexual hormones, and these are also released depending on the different or characteristic that are needed, male pattern or female pattern. For example, female characteristic or male characteristic, these hormones are released. These are also part of your endocrine system. Okay, so they are saying nervous system and endocrine system. This is you could see one is exactly opposite, makeup of the neurons, and this make of the secretory cells, which is the glands. Or information are transmitted in the form of electrical impulse, but it's not electrical, but in the form of chemicals. Okay, so you can easily compare. If you remember one, you can easily compare. One is impulse, another is chemical. For impulse are transmitted along the nerve fibers, okay, axon and dendrons, but chemicals are carried in the blood. As I mean, it's not electrical. This is electrical impulse. This is non-electrical. This is chemical mediator, and these chemicals are released in the blood, and then they are carried to different organs where they can show the effect. This impulse is very quickly, and this is very, very slow. It's not very slow, but yes. Again, you remember, even these chemicals, for example, in case of fight and flight mechanism, is under the control of brain. Okay, so the brain would immediately send signal to adrenaline. Okay, so adrenaline are released in the blood, and then immediately it will increase the heart rate. But if the signals are not sent or delayed to the adrenal gland, so you may not be prepared for fight and flight mechanism. Okay, so don't confuse that. This part is for the electrical impulse is very quick, and the other part is comparatively slow. Okay, you could also see the effect of the hormones maybe last longer, like growth hormones, for example. Adrenaline might work for times, and this may be a little shorter immediately. I mean, the reflex arc, you can easily see immediately you remove the hand, so it will stop. The further impulse will be stopped because you already withdraw your hand, and it's no more needed. But in case of hormone, it may remain for longer in the blood. They are chemical. They need to be metabolized and excreted, So that's why you might be little longer if you compare with the nervous activity. And these are transmitters are really very precious as well. So you want to save them, and they are regenerated. You have a good amount and is stored. Although we do have good physical storage of transmitters, but yes, we save more because they are related to the higher center. So I mean, something which you call the uninterrupted. You don't want any interruption in this supply, because it might be dangerous if your body is not reacting to something in environment immediately. So that could be dangerous. Okay. And I think we have discussed also about the coordination in plants. And one of the mechanisms that we mentioned is a hormone, auxin. Auxin is the hormone that is, and we mentioned about the gravitropism and phototropism, how they are responding to light and how they are responding to gravity, right? So auxins are the one that are, uh, if it is straight and you have enough exposure of light, you might see that there's a linear growth, straight growth. It's not bending, okay? But if you're putting light on one side, okay, for example, this is the direction of light, okay? So this part is not exposed. So this part, auxins are already destroyed, okay? You don't have much auxin here, you have more auxin here. Auxin is increasing the growth at one side, okay? This part is increasing faster, quicker compared to this part, which is exposed to light. So this is why auxin would, uh, this will cause bending towards the light, because the part which is away from light, a shady area, has more auxins, okay? And that's why they are bending towards light. And they have different way how auxins are working. This is one of the things, the effect of light. Or you cut off the auxin supply, you put up some material that can still diffuse auxin, okay? So this auxin, if it is diffusing, you might see the effect of auxin. If you put some material which is not permeable or which not diffuse auxin, like for example mica substance which is may not permeate, you may not see the response of light because the auxins are not basically diffusing. While here you have diffuse agar jelly that can diffuse auxin. That's why this part is responding, okay? And I think we have discussed this part. So this is a gravitropism because the lower part will have more auxin, okay? So the lower part, this is towards the gravitational pull. This is a gravitropism because auxin will be more concentrated in the lower region, and that's why it will bend. If you don't put it straight, if you put the plant horizontal, you will have more auxin in the lower regions, okay? But it is exactly opposite in case of the roots. We call it radicals. Always it will go even in whatever direction you put it. So it will definitely cause the bending. So it's exactly opposite. It's not, although this is a gravitropism, but they are not promoting the growth. The growth will be on the other side, okay? So that's why this part is less auxin, but it's growing more. So this is exactly opposite to what we have discussed in the stems or in the branches, okay? So if you are putting it straight, the auxins are equally distributed. So I think this is something that they have discussed. They have mentioned different experiments here. I don't think there's anything special here that we should focus on. But don't forget gravitropism and phototropism. So phototropism is also related to the light, and gravity is related to gravity. And radicals are always, radical mean the roots, and the roots, the upper region. Although in this region you have more auxin, but these auxins are different in that we have seen in case of stem, okay? That's why you have bending of roots always towards the earth, towards the ground. And this is how it is needed, because the seed definitely will need develop roots so that it can support the shoots and the leaves, okay? So I'm not sure what else we have. Let me see. And the homeostasis, I think then we will close it. Homeostasis future, I think few concepts we discussed before. Homeostasis, I think you know what is homeostasis. To maintain your body temperature, your internal environment constant and stable. Okay, your blood glucose level, your blood pressure, your heart rate, for example, your body temperature. So this is why we have discussed cold-blooded or warm-blooded animal, those who can respond to their environment or they don't. So remember, we mentioned this iguana is depending on the heat source outside, not like our body, or it cannot respond the way we are responding. Our body is responding to heat very easily, okay? So and then you have already I think mentioned here, or endothermic or exothermic. What what we have discussed, the main part in our body that we have discussed was the skin, you remember? And the major part was the dermis. Dermis is guard most of the things. You have sweat glands, you have the piloerectic muscles that are erecting the hair on your skin, and also the sweat glands you could see here is following maintaining the body temperature. So this is, your skin is an efficient example of homeostasis. You remember when it is hot weather, you could see this mechanism we discussed before. When the body is too cold, okay, so when the body is too cold, what will happen when the body is too hot? So you can compare these two. And I think in some books they have given an example that they have put the vessel little bit down the skin, okay? So remember that these vessels are at the same place. They are not moving up and down. So don't confuse. If you have any option with the space between these vessels and the upper layer of the skin is increasing or decreasing, that is not an option. That's absolutely wrong. Scientifically, that is not correct. How your vessels are responding, they're responding by either vasoconstriction or vasodilation. You know, in case of if the body is too cold, so what happens to the vessels? See here, they're constricted, okay? And your piloerector, the hair are also straight because it can trap air, and air is a bad conductor. If body is too cold, you don't want to lose heat, okay? So this is one of the things that your body is preserving heat. That's why capillaries are constricted, so you have less blood supply to the skin, less sweating, and more air is trapped here, okay? And that's why you have good preservation or good control. You don't want to lose heat, so you're preserving heat, okay? Well, when it's too hard, you want to get rid of heat. That's why your vessels are dilated, okay? These vessels are dilated here. You have more exposure and more heat. That's why in hot weather, you might see under the skin, the vessels are dilated. It's very easy to see that. But in very cold weather, your skin is pale, okay? And you won't see vessels. It's hard to even draw blood. If you want to withdraw blood from the vessel, it's very hard to take it. But in case of very hard weather, you have sweating also, and you don't want piloerector muscle to be contracted because you don't want to trap air here also, okay? So if you're given these two diagrams, and you might be asked question in different way, but always understand the basic principles, okay? Basic principles is that if your body is too cold, you don't want to lose heat, and if your body is too hot, you want to get rid of extra heat, because you want, this is called homeostasis, you have to control normal temperature, right? So this is how your body is responding. So I think they have mentioned the same thing here. The hypothalamus is also controlling your body temperature, so it is responding. Your blood is too cold when it's passing through the hypothalamus, is it cold or warm? So that will, depending on it will send signal, you know, how to control, either to dilate these vessels or to produce more sweating or less sweating, okay? So let me see what else we have. I think they are again explain the same thing in different mechanism. Okay, who is outside the meeting?
Sir, sorry to interrupt, but approximately how much time is left?
37. Your body is maturing up to 37, right?
No, I'm asking that how much time is left in the class?
Okay, I think we have to finish maybe in 5 minutes. It's already 9 o'clock.
Okay.
But I think one concept, this we have, I think this is the same thing we have discussed. Just one thing, I received some question about this aspect, so I will discuss diabetes and then I will close it, okay? So but those who want to leave because tomorrow you have exam, we better to finish it quickly. So this part, some students ask in the group as well that we need to explain what exactly is diabetes. But this recording will be uploaded, inshallah. You could see if you want. So diabetes, you know, is your blood glucose level is not controlled, okay? Muhammad, do you have some question? Okay, so remember that two things are very important in diabetes. We call it diabetes type 1 or type
Sir?
2. Yes?
So will we do chapter number 16 also?
Chapter number 16. What exactly is that chapter? I forgot the
Like
name.
reproduction of plants.
Okay, okay. Let me finish this. And those who want to stay, okay, I'll try my best, inshallah, to cover that. So diabetes, you remember, is the, keep in mind, type 1 diabetes. When we say type 1 diabetes, what happened is, as you mentioned, that when you're eating, you're taking meal, you are eating sweet things, you have glucose in the blood. So how this glucose, how your body is responding, your body will secrete insulin. Insulin is a hormone which is taking care of the glucose, right? So it is storing glucose in the in the liver. It's also by your uh, cells, so you can use some part of the glucose for the energy and also for the survival, for different reaction that we need. This is called the oxidation of glucose and oxygen. You produce more energy, and insulin is released. But what happened in some people, there is a problem in pancreas cells are damaged. Okay, pancreas cannot release insulin. So if there is no enough insulin, people will have insulin injection. You remember, they take insulin injection to get control because their pancreas is not responding, and they are different immune system disease. Maybe their own body system has damaged the pancreas, and that's why they have less insulin or not insulin at all, and they would need insulin from outside source. Some people are taking anti-diabetic drugs, okay, the tablets. They do have insulin, but it's not enough. So just they need little push. But in type, that is called type 2 diabetes. Okay, type 1 is the one that here have no insulin at all. Okay, so when a person is eating meals which is containing more carbohydrates, so it is basically triggering the release of insulin because you have more carbohydrates. You have, so your body is receiving more glucose now, so you should have more insulin in the blood. So this blood glucose level will drop. Okay, uh, and they have mentioned here, I think, at one hand, if you are eating too much carbohydrate, that will lead to hyperglycemia. And if you don't have insulin and you have high glucose level, high glucose in your blood, that's called hyperglycemia. Okay, but if you are not eating carbohydrates, okay, and of course your insulin is also less. Insulin has not stored glycogen, because even if you have less carbohydrate, you still have glycogen stored in the liver, so that will release glucose. But in case of diabetes, of course, they don't have much glycogen in the liver because there was no insulin, right? So they will develop hypoglycemia. Hypo mean decrease. Hypo mean decrease than normal. And this is how they have mentioned that you have to have a good control either on the carbohydrates intake, and that's why if you have hypoglycemia on the higher center, the behavior will be less really abnormal because your body cannot work without glucose. There's called bizarre behavior. They're confused and they have tachycardia and nervousness, and some they call it hypoglycemic shock. People will become unconscious. They can't respond. They cannot maintain their posture. They cannot maintain their activity. They will fall down. So this, because of hypoglycemic shock, both are dangerous. Hyper and hypo both are dangerous. Okay, when you are hyperglycemia, you have more glucose now in the urine. You have more concentrated urine, and you are losing more water. That's why you will feeling very thirsty, and you have dry mouth as well, and you will have more desire to eat carbohydrates because you cannot manage these carbohydrates very easily. You don't have insulin. Okay, diabetic people do not have insulin in their body, so they are at either risk. If they eat too much carbohydrate, they will develop hyperglycemia. If they don't eat well, they will hypoglycemia. That's why these people cannot survive without drugs. They will be given insulin if they are type 1 diabetes, where there is low insulin level. Okay, and then you can measure the glucose either by glucometer. So these glucometer are glucose strip actually. So what happens, you just give a small prick, and this prick will drop of blood. So this glucometer can read actually. They have a stick. When you insert the stick and you put a drop here, so this drop, just by whisking like capillary action, this will immediately pick up this small amount of blood. So it can easily detect the amount of glucose in your blood. This is called glucometer. Okay, so either to check in the blood or to check in the in the urine. Okay, and the urine, these are called urine dipsticks, and this will also give you glucose. Ideally, you should not have glucose in the urine. Diabetic people have a lot of glucose in their urine. So when you take this sample, urine sample, you have a stick, you dip it, and this will give you different colors. So it probably may not give you exact idea how much amount, but yes, it can tell you that this is abnormal, that the person has got more glucose in the urine. Although we have different tests available now, either you measure it by blood or you measure it by in the urine, because as you remember, the glucose is filtered and then it's reabsorbed. And diabetic people, there's so much load on the kidney, so they cannot take care of the excessive glucose which is filtered in the urine. Okay, so that's why you will see more glucose in the urine. So I think drugs we have covered. So those who want to take a little rest, I think they definitely can go, and inshallah I will post this recording for sure for you guys. But I think you're also asking about the reproduction, and we mentioned about the reproduction in this chapter, asexual reproduction and sexual reproduction. Okay, when we say asexual reproduction, that's exactly the same parent, one parent only, like in case of banana, or for example, potato tuber, right? So this potato tuber, you just cut it, and then you plant it somewhere else, you can exactly get the same plant. Okay, you are not producing or growing this plant from the seeds. The seeds that are we have, that's why in banana these days we don't have seeds, and also in other oranges and many food, they are genetic basically. So they are growing it from the same parent, one parent. Okay, when we say sexual, there has to be both male and female gametes. Okay, like we have eggs and sperm, for example, and then we have pollen grains and ovules in case of plants. We'll talk about some of these things. I think in the sexual reproduction, we call it gametes are called sex cells. Okay, so male gametes are called sperms, for example, in humans, and female gametes, these cells are called egg cell, for example. Okay, or ovum. And you remember that in case of can I have water? Yeah, in case of what do you call it? In case of asexual reproduction, remember the number of chromosomes are fixed. Okay, they have all 23 pairs, for example, in our cells, right? But in case of gametes, where there is a fusion of male and female cells, sex cells, so you are receiving half from each parent, half of it. Okay, half of genetic material from, for example, when egg and sperm are fused, so you're making zygote. Okay, same is the case with the pollen grains. Pollen grains we discussed in the plants, when they are transferred to the stigma of the ovary. So that is, the nuclei is transferred and is fused. I think we'll talk about just in a while. But I think this is the thing that we call it the male body cells, and we call it meiosis. So these are 46 chromosomes that are now providing 23 chromosomes. Okay, this is one part of, in our body we have sex cells. So XY, for example. Okay, XY in case of male and X in case of females. So these are called diploid. Okay, these are called haploid. Haploid means half, and this is diploid. Diploid means the normal number of the chromosomes. Okay, so this is related. These are mitosis. The cells that are dividing by mitosis have got diploid number of chromosomes. Okay, diploid number of chromosomes, and the one that is half, and that's called haploid number of cells. And these are in sex cells, and we also call it gametes. Okay, so and when these gametes are fusing of male and female gametes, is producing zygote. So now zygote is receiving 23 from here, for example, 23 from here. So you have again 46. So this cells is now dividing. This has got all the genetic material to grow further, multiple division finally into organisms, right? So we have been created from single cell. Okay, that's why Allah subhanahu wa ta'ala is mentioning that we are worth to mention. Nobody knew about this whole physiology and whole genetic biology, right, and molecular biology. And Allah subhanahu wa ta'ala specifically mentioned in Quran, many verses of Quran deal with this beautiful embryology and all these developmental process. Okay, so let me see what we have here in the plants. I think that was the most important that we have in the plants. We remember the flowering plants. So you have this part, which is the male part. We call it stem or anthers. Okay, and this filament is holding this anthers. This anther has got pollen grains, right? So this part is called the carpel. So you have style, and then you have ovary that has got ovules. Okay, so pollen grains are transferring. This transfer of pollen grains to the stigma of the carpel, this of the ovary, is called, what do you call it? Pollination, right? Do you agree with that? This is called pollination. So flowering plants, remember, flower is the sexual part of the plant. Okay, and that is propagated in the sexual reproduction. Why? Because it will produce seed. Okay, that's why flowers, definitely this transfer is not automatic, right? These flowers have attractive colors and even different shapes and different ways how to transfer pollen grains from anthers to the stigma, and the stigma finally is pollen grains are developing tubes that will transfer nucleus. So this is also and they have mentioned now further that how pollination will take place, because this is needed now. Male and female fusion is needed. So plant has flowers, attractive color, and also nectars. Okay, this is a sweetie and gummy materials that many insects would come and they will sit on it, and the pollen grain will stick to it. And then when they go from one plant to another, so they transfer these pollen grains to the stigma and finally causing pollination and fertilization. Okay, so after pollination is taken place, and they say the carpel, which is they have described these different parts. The ovary, inside the ovary there are ovules which may contain female gametes, and the pollen grains are then producing transferring this nuclei to these female gametes, which is ovules, right? And that's you can have multiple ovules. That's why you can see there are multiple ovules, and that if that is transferred, you have multiple seeds also. Okay, so the ovary part is fruit basically, and the inner part will be generating the seeds. Okay, and then you have protective things. All these are just protective uh, to protect the petals and protect also the part uh, which is participating, those parts that are participating in pollination and also in the reproduction, because that will generate seeds. Okay, and then they have described pollen grains and ovules. I think I've discussed about the pollen grains. You can just go through it. It's nothing to be very complicated, but it's extremely simple. I just mentioned that here. Let me see. And then they have mentioned the pollination. What are different processes of pollination? So I think pollination, then there are different types of pollination. It's by wind or by insects or cross pollination or self pollination. So as you remember that if we the pollen grains are transferred from one flower to the plant of another flower, that is called cross pollination. Okay, and then you have self pollination. If it is deposited, the pollen grains are deposited on the same flower. Okay, these pollen grains are falling on the same flower, so that is called the self pollination. So these are different, I think, pollination activities. And then they have mentioned like what kind different shapes of the flowers. As you mentioned, the wind pollination has a very special structure. You could see here that these are large and hanging out. Okay, so these can easily trap these uh, uh, the pollen grains can be easily trapped, and you could see here the anthers are also hanging. So you could easily compare those with the insect pollinated or wind pollinated. For example, in desert, you may not see a lot of insects and birds, so they are mostly wind pollinated, and very rarely also water pollination. These are also going through water, some of the plants. So they have, I think, they have compared these wind and insect pollinated and how they are very prominent and very good and attractive and scented, and nectaries are there, the petals are there, and the filaments are inside and they are dangling. You could see here as hanging outside, so they can easily trap the pollen grains that are coming. And I think once actually the pollination has taken place, you remember that we mentioned what are different kind of reproduction, sexual or asexual. Asexual in the banana plant, in potato tubers that we mentioned, and also we cut branches and then plant it, okay, different areas. So there is also asexual reproduction. In case of sexual reproduction, we are talking about gametes, okay, egg cells and also sperm and egg cell, or in case of plants, pollen grains and also the ovules here. So once they are thick, the pollen grains are transferred to the stigma, which is sticky material, okay. It can stick the pollen grains. So and then they are developing tubes, right? So they are growing these tubes, and the pollen grains are transferring this nucleus to micropyle. And this micropyle, you can see here, this is a small hole. There will also remain even in the seed, if you see it, and that will remain, that can absorb water for the seed. So you could see that when it is fused with the ovules here, so now that is called the zygote. The seed formation will take place. So the external part will form in the form of ovary. You still see this part of stigma, and you could also see seeds, okay. So once the seeds have been developed, so you can store these seeds. The seeds definitely should not contain any water. It is cotyledons, which is full of starch and other enzymes, but these are making as an energy reserve, okay. They are storing energy for the seeds, so that it can grow. Initially, if you put this seed, plant the seed, even if you don't have much water or much sunlight, of course it is underground, it will still have enough energy for the shoots to come in, these seedlings or the new plants will develop, okay. And then you have also different I think we have mentioned the scar where this was attached here, so there's called the hilum. And then you have the testa. This is a very water resistant protective covering of the seeds. And then you have the plumule, which is the embryo itself, okay, for this one to give you the shoots also. And then you have the cotyledons also. So this part is producing radicle, okay. So the radicle are the roots, okay, and the plumule part is called the shoots, okay. And together this is called embryo part of the seed. So you might see that micropyle that was here is absorbing water, because you need some water so that the seeds can swell. And for the seeds to be good enough, should not be eaten up by, of course, by pest or insect, and it can easily be destroyed also if there's some water contents in it. The seeds are usually dry, so these dry seeds can stay longer. That's why when you want to grow them, you need to give them some water. That's why in rainy season people will grow crops, wheat crop, right? So this the micropyle will absorb water, and now these seeds are no more dormant. So the cotyledon part, which is mostly starch, and the enzyme will start working, and it will generate more energy. Okay. So I think that's it, probably. And you could see here how their seeds are generating. You could see this roots part, even the testa will move away. When it will start, the cotyledon will also turn green, because now it will take part in the photosynthesis. So till here, uh, when it's growing, remember that it's the stored energy in the seed, and we call this germination. So remember, for germination you need good temperature, of course you need some moisture as well. Okay, initially you may not need light, but yes, of course later on definitely you need light. That's why in the forest some seeds will germinate, but the plant may not grow well because they don't receive enough sunlight, okay. So they will die. They are definitely are going very fast and very tall, but they are very thin and pale because they cannot carry on photosynthesis. So germination, of course, then finally will produce a new plant, okay. So this is the final step where seed germination will take place. I think that's it. Probably you have tomorrow inshallah exam, and if you have any question related, otherwise we'll stop it here, and then you go through some of the concepts we discussed here. Okay.
So can you do the experiment question?
What is the experiment question? This part.
Yes, sir.
Okay, this is the, okay. So what exactly is showing? Just okay. These are the tomato seeds they are saying, and they say wet cotton wool, and this is a perforated zinc platform, and here is water. Okay, and here is a cotton wool. Okay, so there, I think what exactly they are showing is the germination of seed. They want to see what conditions are basically needed for this to grow. So tomato seed, for example, and you have all the water here also, and here is a warm. What is this? Okay, this is in warm, light place. Okay, so you have probably good condition for growth to see how. Now compare this with part B. You have same water, but this is in cold and dark place. Okay, you have wet cotton, which is almost the same. All the seeds are the same, and you also put a cotton wool here just to plug it to prevent it from the external environment, but you still have porous way to have some contact with external air. And here is part C in warm but in dark place. So this is warm in light, this is warm in dark, this is in cold and dark place. Okay, so now in the warm place, this is again a warm place, this is wet cotton wool, and this is the pyrogalol. I think in sodium hydrochloride solution. Okay, this chemical I've not read it, but let's see. Okay, we will see the results. Okay, so here's a dry cotton wool, and here's a in warm light place. Okay, so now they are asking this question, light. What are the three? Can you put the tube A, D, E in warm place, in a laboratory in the light, and put tube B in a refrigerator. Okay, tube B in the refrigerator, and put tube C in a warm, dark cupboard. Now, what are the three conditions the tomato seeds need for the germination? Explain. Okay, now there are again different condition. If you remember, we did one experiment about the mustard seed. So what exactly is needed? So probably you don't need light initially for the germination, right? The one thing that we mentioned here in this diagram, you didn't need light initially because the seed is basically covered and it will germinate, of course. So in case of cold and dark place, so I'm not sure if this they have given a figure very clearly. So what do you see? Where exactly the seed will germinate? In which condition fulfills all the required conditions where the seed will germinate? Do you think? So this is not providing a forced water, and you can see it does not provide the ideal environment for it, because this is a cotton wool, so you don't have an external environment, and it does not provide moisture as well, right? So this the condition is mostly tomato water, and this is wet and also a little bit warm place. So this is the best environment for the seed to grow. And I think they have mentioned this condition. I don't know if we have discussed it here or not. Let me see. Okay, let me see the question because we have short of time instead of just. Oh, so explain why each of these condition is needed for successful germination. I think this is again, this experiment is related to what we have discussed in the seed germination, the condition that you need here. What exactly is needed for the germination of seed. Did we mention here? Yeah. Okay. Where is the germination? Let me focus here. Okay. Yeah, this is the seed germination. Okay. So now you could see here, without water there will be no metabolism. Moisture, okay, some water, okay, because germination definitely would, if you remove water, of course, dehydrated, so seed will not germinate because no metabolic reaction will take place. The seed will be inactive or dormant. Okay, a seed must be in a certain condition before it will begin to germinate. You can find out what they all are. An activity when a seed germinate is first take up water through the micropyle, is the water goes into the cotyledon. So first thing is the moisture, they will swell up and eventually that will burst, okay? Once there's a sufficient water, the enzyme in the cotyledon become active. For example, is okay, and then you have proteases, okay, maltose, and all these are producing energy. So they diffuse soluble, yeah, okay. So what basically in this experiment they are showing that you should provide enough water, for example, moisture, and also good temperature. If you are putting this in extreme cold water, especially this one, this will not grow. Why? Because through the micropyle, if the water will penetrate, so it will definitely activate the enzymes, right? So these enzymes are providing energy, initial energy for the germination, okay? But this is not going to take place in a dry place, okay, or in a cold place. Where a cold place, for example, in a refrigerator. Do you think in a refrigerator these enzymes, even if you have water, will it grow? These seeds will grow? Will it grow or not? Will it not grow? Any explanation?
It will grow.
Why it will grow? In
Sir,
cold
because
place.
that cotton wool and water and oxygen is
Yeah,
also there.
So it will not, maybe.
it will not, because enzymes, definitely the enzyme reaction will take place at certain temperature. If it is really too cold, that is why some plant, of course, like that's why they don't grow in winter. They have different season, yes. But if you put in a refrigerator, which is around 4 degrees Celsius, you may not see that they will grow. This, especially tomato seed, will not grow in a refrigerator because the cotyledon, the enzyme which are there in the cotyledon will not be active. They will be dormant until you put it in a warm place, okay? So this is really is not going to grow. So because you don't have any enzymatic reaction, you are providing water, but you are not giving ideal environment, okay? So light is probably not a big deal. As I mentioned, that it will still grow, of course. If you remember in the mustard seed experiment, I think we did it in a question probably. And this one is not going to grow because a wet condition, and it's not providing enough. This is also kind of exactly, I didn't read this very carefully. I'm not sure about it, but this is depriving actually the moisture or the wet environment. So although this is in a warm place, but it's still not going to grow. So these are not fulfilling the ideal environment, okay? These two especially, okay? And including this one also. They are also providing good environment, but except the temperature is not there. So temperature is needed. Light may not be a big issue, and moisture is needed, and good temperature is needed. And of course, you need to, when there is a growth will take place, the germination, you also need oxygen or supply. That will also be needed for the germination and for the photosynthesis to later on carry on in growing the plant material. And I will read it somehow to see what exactly detail they have given, the other details, because I have not focused this on before. If remember, we didn't cover it. But they have mentioned here that yeah, paragalol is absorbing oxygen, so it's not providing much oxygen here. So this may not be one of the things that uh, is really not good environment. Your teacher will handle it for you because you should not use it yourself. Set up these tubes showing the diagram, and the paragalolol is absorbing oxygen, so oxygen is deprived. Do you think initially oxygen will be needed for the seed germination? Do you think we need it?
No.
No, initially it may not be needed, right? So initially it may not be needed for the oxygen generation, yeah, for the germination of seed, because even if you don't have air contact, you still will see the seed germination, okay? The only thing would be, yes, the dry cotton. So this is without any moisture. So if there is no moisture, there will be no activation of the enzyme. You may add some more activities to read on it. to because I haven't actually read this experiment, but I can add on some of the thing if it is needed. Let me, maybe I can post in the group the more explanation if you want it. But is it included in your course? This is included?
Yes, sir.
Okay. If this is included, inshallah, I will post the voice message explanation inshallah in the group, so you can get more information there. Okay.
Okay.
Is there anything else you want me to cover? I think we are done. You need to take good rest because you have to get up early morning, right?
Yes.
Inshallah. If there is anything, I'll post this recording as well, and then I will try my best to explain few things that are needed. I wish you all the best for your exam, inshallah. Assalamu alaikum wa rahmatullahi wa barakatuh.
Walaikum assalam, sir.
Thank you, sir.
Thank you, sir.
My
Thank you,
pleasure.
sir.
Welcome.
Allah Hafiz, sir.
Thank you, sir. Thank you.
Uh,
Hafiz. Assalamu alaikum. Yeah, assalamu alaikum wa rahmatullahi.